Hello friends, welcome to Unique Classes. In this video, we are discussing about cell cycle. The cell cycle is the series of events that cells go through as they grow and divided. It consisting of several stages categorized into two broad phases, interphase and the mitotic phase. First one is interphase. Interphase is the phase where the cell prepare for cell division. It is divided into three substages. First one is G1 phase. Duration of G1 phase varies in length, can be short or extended. Characteristics of G1 phase. Cell grows in size, produced RNA and synthesized proteins. It is a period of intensive biochemical activity. Second stage was S phase or synthesis phase. Characteristics of S phase. DNA replication occurs each chromosome duplication to form two sister chromatids connected at the centromere. Outcome of S phase. Each cell will have twice the number of chromosomes, but they are still in the form of chromatid pairs. Third stage was G2 phase. Characteristics of G2 phase. The cell continues to grow and produce proteins, particularly those needed for mitosis. Organelles are duplicated and the cell prepare for the mitotic phase. Second phase was mitotic phase. The M phase is where cell division occurs and it is divided into two main processes, mitosis and cytokinesis. Mitosis, the cell was divided into two phases. Mitosis have four substages. First one is prophase. Chromatin condensed into visible chromosomes, the nuclear envelope, breakdown and the mitotic spindle. Second phase was metaphase. Chromosomes align at the cell's equatorial plane, metaphase plate, and spindle fibers attach to the centromeres. Anaphase. Sister chromatids are pulled apart towards opposite poles of the cell. Last one is telophase. Chromatids reach the poles, decondense back into chromatin. The nuclear envelope reform around each set of chromosomes and the spindle apparatus disappear. Cytokinesis. Characteristics of cytokinesis. Division of the cytoplasm and organelles into two daughter cells in an animal cells. It involves in the formation of a cleavage furrow will in plant cells. A cell plate form. Cell cycle regulation. The cell cycle is tightly regulated by checkpoints and regulatory proteins. First one is checkpoints. G1 checkpoints determines if conditions are favorable for cell division, checks for DNA damage and cellular nutrients. G2 checkpoints ensure DNA replication is complete and checks for DNA damage. M checkpoints are spindle checkpoints. Verify that all chromosomes are properly attached to the spindle apparatus before anaphase begins. Regulatory proteins. Cyclins, protein that regulate the timing of the cell cycle in eukaryotic cells. Levels of fluctuate. Regulatory proteins, cyclins, proteins that regulate the timing of the cell cycle in eukaryotic cells. Levels of fluctuate throughout the cycle. Cyclin-dependent kinases, enzymes that when bonded in cyclins phosphoglycerate target proteins to drive the cell cycle forward. Tumor suppressors, such as a p53 which can help the cell cycle in response to DNA damage and RB, which regulate the annexation from G1 to S phase. Oncogenes, mutated genes that drive the cell cycle uncontrollably and associated with cancer. Thank you for watching. Like, share, subscribe to my channel.